So I'm very pleased that we have Rebecca Ashare here. She's assistant professor in the Department of Psychiatry at the Perlman School of Medicine at the University of Pennsylvania. She received her Ph.D. in clinical psychology at the State University of New York in Buffalo in 2011 and completed her postdoctoral clinical fellowship at Yale University School of Medicine, Division of Substance Abuse. She then joined the Center for Interdisciplinary Research on Nicotine Addiction at the University of Pennsylvania as a postdoctoral fellow and was appointed to faculty in 2012. At the University of Pennsylvania, she established a program of translational research related to health behaviors. Her work focused on identifying novel therapeutic targets with a focus on cognitive control, decision-making, and stress that may represent risk factors for smoking relapse, and evaluating novel treatments to improve abstinence rates. Her research leverages tools from the fields of psychology, neuropharmacology, and cognitive neuroscience to understand the mechanisms of efficacy of novel interventions. More recently, her work has focused on identifying unique risk factors for smokers with comorbid health conditions, particularly those with HIV. Dr. Ashare is currently the recipient of 4 funded awards, 2 R01s, a K23 Early Career Development Award, and an R21, and collaborates on several NIH-funded grants evaluating multiple health-related behaviors. She's also a senior fellow in the Center for Public Health Initiatives at the University of Pennsylvania and teaches core courses in the Masters of Public Health program. I'm very pleased that she's here, and I'm very glad that we're going to be hearing her research. So thank you very much.
Thank you for having me, Warren. Oh, I have to turn my mic on, sorry. All right, can everybody hear me? Okay. Thank you all for coming this morning. I had a chance to meet some of you yesterday during my visit, and it's been a really fun visit so far. So I'm excited to present some of the work that I've been working on for the past 5 or 6 years. And if you have questions at any time, feel free to interrupt me. This doesn't have to be me just talking at you. I'm very willing to answer questions throughout. So as Warren mentioned, my work over the past few years, we really do take a translational approach to our science. And so what do we mean by that? And so we leverage tools from preclinical and basic science, neuropharmacology, genetics, and neuroscience to really identify what are some of the risk factors that are associated with in particular smoking behavior or eating behavior, the health behaviors that we're very interested in changing. And we use those tools to identify novel therapeutic targets that then we can evaluate and develop novel interventions to then target those behaviors. And I'll be talking specifically about a couple of different models that we use to evaluate those interventions that we think are efficient ways of screening these interventions that can help us detect early signals for efficacy. It's kind of a quick win, fast fail approach, so that before we invest a lot of resources in a large clinical trial, we have some sense of whether this might be efficacious. And then the last piece of this is that we know there's never going to be a one-size-fits-all treatment. Our treatments are going to be more effective for some people than others, and so I'm very interested in understanding how can we identify which populations will benefit most from a particular treatment, and what are the measures that we can use to identify them. And so this work that I'll be talking about today will focus on 3 primary areas of research. The first is on identifying cognitive factors and decision-making during nicotine withdrawal. And for that, we use neuroscience and behavioral tools to identify those factors. The second set of studies is focusing on these novel interventions and different screening tools that we use to evaluate these interventions. One of those studies will focus on smoking behavior. The other one will focus on eating behavior. And the last set is kind of a new area of research for me that's evolved over the last couple of years, focusing on the intersection of HIV, cognition, and smoking, and why it's important that we target smoking behavior in this population. Okay, so before I go any further though, I have to say, so none of this work happens in a vacuum. All of the people that you see up here are very involved. I collaborate with a great group of people that makes this work possible. These are folks who are doing clinical research, neuroscience research, preclinical investigators, our clinical investigators and study physician, Karen Lerman, who is my postdoctoral mentor, was sort of very significant in my in some of the early work that I've done. And then other folks who are involved in the HIV research and clinical work, and then Joe Cable and James Lawhead, who are our neuroimaging folks who have assisted with a lot of this work as well. And I think that's one of the things that's really exciting about the idea of being here too, is everybody seems really interested in collaborating, and I think that's what makes the science as good as it is. Okay, so why do we care about lifestyle factors? The 7 out of 10 deaths are associated with things like chronic disease, things that are preventable, things that we can address through behavior change. And the other important factor that a lot of other folks, including our legislators, care about is it accounts for about 86% of healthcare costs. And that's, direct healthcare costs, but also costs due to, inability to work, missing work. So there's a lot of factors that contribute to this. And there's a lot of behaviors that we can change, right? So 18%, despite the progress we've made in treating smoking and other public policy, 18% of adults still smoke. Rates are higher depending on where you are in the country. Only 1 in 5 people get the recommended amount of physical activity that are sort of associated with health. And about 40% of people only get 1 serving of fruit or vegetable a day. 86 million people are considered pre-diabetic. Over 1/3 of people are not getting enough sleep, and sleep is associated with all sorts of chronic conditions. A lot of us researchers probably fall into this category. Over 15 million people have an alcohol use disorder, and this statistic was very staggering to me. About 75% of doctor's visits are due to stress-related complaints. And so that's a staggering statistic to me. So these are all behaviors that are associated with lots of chronic conditions that we as behavioral scientists can address. And so again, as I mentioned, despite the progress we've made with smoking, it still remains a significant problem. And so why do we care about it? So it accounts for nearly 500 million deaths in the United States, and accounts for over 1/3 of cancer-related deaths and significant healthcare costs. And importantly, we see a lot of health disparities in terms of the rates of smoking across different populations. So this dashed line here, this is that 18% in the general population. Individuals with a mental health comorbidity smoke at a rate between 30 and 40%. If you have a mental health comorbidity and you're living below the poverty line, your rates of smoking are even higher. And then individuals with HIV and AIDS, their rates of smoking are upwards of between 50 and 74%. So there are populations who are significantly more likely to smoke and are receiving less attention. So there's a lot of health disparities in this area. Okay, so when we talk about our translational model of how we address these health behaviors, so ultimately what we want is we want our interventions to affect our behavioral endpoint, whether it's smoking cessation or making better food choices. And we have a lot of tools that we use to do that. I'll be talking a little bit about some genetics work that we've done to identify particular genetic factors that are associated either with relapse or relapse to smoking, and also genetic factors that can moderate the effects of our interventions on our outcomes. Also talk about some preclinical work that for individuals that we've collaborated with that have served as the foundation for some of the work that we've done and suggested that this is a novel target that we should pursue in human clinical studies. And then I'll talk about some of the cognitive and neural mechanisms that we've investigated to understand how our interventions are working and how we can sort of push those systems to improve our interventions. Okay, so the first set of studies we'll be talking about. So again, why is health behavior important? And so this is a little bit of the model in terms of how we think about it. This is very based on a lot of the work that Warren's done, and sort of these two different systems that are not always aligned with one another. And we know that there are these neural and behavioral substrates of self-control processes. And so when you get an increase in one and a decrease in another, that's when you get individuals who are choosing these more immediate, more risky choices. And then that can lead to some of these poor health behavior choices. And we know that there are common underlying neural substrates that are associated with both cognitive function, so executive cognitive function, and with decision-making processes, particularly in the prefrontal cortex. And we also know that if you can improve cognitive function, that you can also push and you can reduce delayed discounting. And so you can reduce the likelihood that somebody's going to choose that immediate choice over a more longer-term reward. So when we talk about targeting cognitive deficits, so very often when people think about nicotine withdrawal, they think about the very common symptoms of irritability, fatigue, negative affect. But it turns out that about 50% of smokers also report cognitive deficits when they quit smoking. And so when we talk about cognitive deficits, it's not that they are, completely functionally impaired and they've forgotten how to tie their shoes. They're subtle deficits. However, they are clinically significant. So we know that these deficits predict relapse. So the individuals who experience the greatest cognitive deficits when they quit are those who are most likely to relapse, and they relapse faster. We also know that efficacious medications for nicotine dependence, things like bupropion and varenicline, can reverse these withdrawal-related deficits. And so in terms of the types of cognitive domains that we're interested in, so these are things like sustained attention, so your ability to focus, sort of remain vigilant and carry a task through to its end. We talk about working memory, your ability to manipulate information as it comes online. So if you think about if you're interested in cooking and you're trying to follow a recipe, trying to think about, okay, I've done this step, what's my next step, and trying to keep track of all of those things as you're doing the cooking, okay? And then the last one is inhibitory control. And so this is something that we're very interested in the context of smoking behavior. So this is your ability to inhibit a prepotent or an ongoing response. So in the context of smoking, we think of this as when a smoker experiences a craving, their prepotent response is reach for the cigarette. And so in this context, is we want, what is your ability to refrain from reaching for that cigarette in a excuse me, in a craving state? Excuse me. And so we also know that there are a lot of individual differences in cognitive function. And so we were interested in understanding the role of prefrontal dopamine in both cognitive function and smoking relapse. And so we became interested in this genetic polymorphism in the catechol-methyltransferase, or the COMT gene. And the part of the reason for this is, there's a lot of different pathways that we could have looked at that are involved in regulating dopaminergic function. But this gene, this genetic polymorphism has demonstrated associations with cognitive function and with the ability to quit smoking. And essentially what it does is that at this particular exon, there's a switch where the VAL allele, so individuals who carry this VAL allele, it increases the rate at which catechol-methyltransferase breaks down prefrontal dopamine. So the VAL allele is more active, so it breaks down more prefrontal dopamine. So individuals that carry one or two VAL alleles essentially have less prefrontal dopamine on board. So they're in a kind of a hypodopaminergic state. And there's been several studies showing that these individuals exhibit deficits in cognitive function. So we know dopamine is important for maintaining cognitive function, and they're more likely to relapse to smoking. And so we were interested in, well, if these folks are predisposed to cognitive function, cognitive deficits, and more likely to relapse, is it possible that these individuals are experiencing greater withdrawal-related deficits, and that is one of the factors that is related to their ability to quit smoking?
Maybe?
Yes.
Quick question. When you mentioned that polymorphism, you talked about differences in prefrontal dopamine. I'm just curious, this is a mutation that's expressed in all areas of the body with respect to dopaminergic synthesis, or is it a location-specific mutation?
So the mutation itself is not location specific. It affects prefrontal dopamine specifically because the concentration of COMT is predominantly in the prefrontal cortex, and so I think that is why it has such a strong effect in prefrontal dopamine levels.
But other areas of the central nervous system that have the same methyltransferase are equally affected?
Correct.
Okay.
Correct, yeah. And so, for example, COMT inhibitors, so medications that inhibit this enzyme, are often used for treating Parkinson's disease. Things like tolcapone are used to increase dopamine levels. So we were interested in whether these individuals would exhibit greater withdrawal-related deficits. And so this was a neuroimaging study, an fMRI study. And we pre-selected smokers for this. So we genotyped everybody before we enrolled them so that we could get equal numbers of individuals who were homozygous for the VAL allele and individuals who are homozygous for the MET allele. And we tested them on a variety of cognitive tests. We were particularly interested in working memory. And we predicted that the VAL group, so the individuals with lower prefrontal dopamine, would have worse working memory performance when they were abstinent compared to when they were smoking, and that they would exhibit reduced brain activity in regions that we know are important for executive function. So we were specifically interested in the bilateral dorsolateral prefrontal cortex and the medial frontal cingulate gyrus. In a second question, we were interested in the role of brain regions that are deactivated when you're doing a cognitive task. So these are regions that are kind of associated with the default mode network, regions that are generally deactivated during goal-directed behavior. There's been evidence to suggest that when these regions are activated when you're trying to engage in a cognitive task, that it can interfere with your ability to complete the task. And so we predicted that they would show less suppression of activation. So it's kind of a double negative. So essentially they would show more activation than when they were smoking than during when they were abstinent. And so this is a paradigm that we've used. It's called our abstinence challenge paradigm. We've used it both in and out of the scanner. The next study I'll show is a behavioral study, so we can use this in and out of the scanner. And essentially what it is we scan everybody twice, once while they're smoking as usual, and once after 24 hours of biochemically confirmed smoking abstinence. And we counterbalance that to control for practice effects while they're in the scanner. And the brain regions that we were interested in, as I mentioned, were the bilateral dorsolateral prefrontal cortex, the medial frontal cingulate gyrus. And the task that they do while they're in the scanner is a working memory NBAC task. And so for anybody who's familiar with this task, forgive me as I go through and sort of explain it, but what they're instructed to do in the NBAC task is, so in the zero back condition, they're essentially said, okay, here's the target. So they might say, every time you see this image, press the wheel. In a one back, they say, every time you see an image that matches the one right before it, that's a target. So then you get all the way up to the three back condition, and you say, now you have to press when the image matches the one that was 3 before it. So you have to keep in mind all of the images as they move along. So you have to say, okay, this one, all right, that's a target because it matches this one that was 3 before it. I am terrible at this task. I can't do a 3 back. I am amazed at some of the performance that we get, some people who can do this. We've actually just started using a 4 back task, and I don't know how people do it. It's remarkable, but they can do it. So these are our behavioral performance measures. So this is reaction time on the task, so how quickly can they respond. Processing speed has been shown to be effective during nicotine withdrawal. And what we see was consistent with our hypothesis. So the VAL group showed an abstinence effect. So in general, they responded more slowly, so they were processing the information more slowly, and they showed an abstinence effect. So they responded more slowly when they were abstinent compared to when they were smoking. When we look at accuracy, so this is just the number that they got correct, we see no abstinence effect whatsoever. And this is kind of a common finding that we've seen in several of our studies where we see an effect on reaction time, but not accuracy. Now, these are our imaging data. So these are the 3 regions in the executive control network that we were interested in. And so the MET group showed the typical pattern. So they show reduced brain activation during abstinence compared to smoking. This is very consistent with many of our other findings. And remember, we were interested in whether the Val-Val group showed larger differences during abstinence. They did not at all. So in general, there was no abstinence effect whatsoever in the Val-Val group. Now, this was intriguing to us because it was contrary to our prediction. We checked the coding of our groups many, many times to make sure everything was coded correctly. But it was a robust finding. So it survived correction for multiple testing. And so what we wanted to know was, I'll come back to that in a second. But it was a robust finding. And so we were really interested in whether this was something we could replicate. So now just to briefly talk about, so these are the 2 regions that we were interested in our default mode network. So these were the regions that we thought we would see less suppression of activation in the Val-Val group during abstinence. And that was, in fact, what we saw. So this was consistent. So we see that during abstinence, there's less suppression compared to when they're smoking, and we see no effect in the MET group. And so this suggested, so this was consistent with our hypothesis. The interaction in the ventromedial prefrontal cortex was not significant. So again, so we wanted to know, can we replicate this finding in the executive control network? It seemed like a robust finding. Is it something that we can see in an independent sample? And in fact, so we had another data set where we had genotypes on everybody. And we had a sample of about 48 smokers, 27 MET carriers, 21 in the Val-Val group. And what was important was these individuals underwent essentially the same procedures. So they completed the identical fractal NBAC task, the same scanning procedures, and so we were able to see, do we see the same effect in that population as we saw with our original finding? And we did, in fact. We found concordant results in the bilateral dorsolateral prefrontal cortex and in the posterior cingulate cortex. And so this gave us much more confidence in our finding. And so, this was a good example of, you don't always find what you think you're going to find, but it doesn't mean it's not real. And so this was an area, and we followed up this study with a study looking at a COMT inhibitor to see if it attenuated nicotine withdrawal during abstinence. So this was a finding that we then translated into a medication study. But I'm not going to present those results because I have lots of other data that I want to show you. So the next set of studies, so in addition to cognition, so we've shown that the bilateral dorsolateral prefrontal cortex is important during nicotine withdrawal. So we see reduced activation during nicotine withdrawal. But the dorsolateral prefrontal cortex is also really important for decision-making processes. And so it shares this common neural substrate. And we know that there's been a lot of work showing what the neural substrates of decision-making are. So we have a good understanding of that. And I think what we don't have a great understanding of is some of the psychological processes or the mechanisms that lead to the types of decisions that we make. And so when we thought about what is decision-making, right, we think about it, or impulsive decision-making. It's an inability to wait, right? We talk about it as acting on the spur of the moment. And so this suggested to us that timing processes are very important in decision-making. And so we were interested in how the perception of time is related to decision-making. And it showed there's evidence to suggest that orientation to the present, so individuals who are very focused on the present orientation, that's associated with impulsive behavior and drug use. And so we hypothesized that this perception that time is passing more slowly, right? So if you're if the perception that time is passing more slowly, and say you're in a state where you're feeling hungry, right? And time feels like it's moving very, very slowly, right? You might feel like you want to alleviate that short-term discomfort, right? So you're going to eat faster. You might make a different food choice if time feels like it's moving more slowly than you would in the name of the maintaining for a long-distance behavior or a long-term goal. And so we conducted 2 studies. And so in the first study that I did, we wanted to know, well, how does nicotine withdrawal affect time perception? Because we hadn't done that study yet. Nobody knew. And so we collected a sample of smokers who were not treatment seekers, and we used our abstinence challenge paradigm. So we had them do a battery of tasks while they were smoking as usual and during a state of abstinence. And in the second study, what we wanted to know is, how does withdrawal-related changes in time perception and decision-making, so how does that change from when somebody's smoking to when they're abstinent? How does that predict the ability to quit smoking? And this is where one of the models that we've used to screen our different interventions. And so we do is everybody does the abstinence challenge paradigm, so they do those 2 sessions, and then everybody undergoes what we call a practice quit attempt. So it's about a week-long period where everybody is asked, just try your best to not smoke during the whole week. We give them small financial incentives to not smoke during that time. And the reason that we do this is because we've shown that the number of days that somebody can stay quit during the first week of a quit attempt is highly, highly predictive of long-term success. So if you relapse within the first 7 days, there is a very strong likelihood that you're at 6 months, you are also going to be smoking. And the likelihood that you are going to remain abstinent at 6 months is strongly related to how well you do during that first week. And so this is a model that we can use to identify predictors of relapse in a more efficient way than following someone for 6 months or a year. Okay, so this is the time perception task that we used. It's a time reproduction task, and it essentially has 2 stages. So the first stage is this encoding phase, where we put up this stimulus on the screen for a period of time, and they're asked to think about how long this image is on the screen for. And then during the reproduction phase, they essentially have to reproduce the period of time that they thought the red cross was on the screen. Okay, so this is an index of how much time do you think passed during this time window. And the outcome that we use is theta, and it's a ratio of the reproduced duration to the actual duration. So a value greater than 1 indicates that you were overestimating time, and a value less than 1 indicates that you were underestimating time. And so in the first study, we saw that there was a significant effect of abstinence. So when people are smoking, they're pretty good at figuring out how long time, they're pretty good at time perception, which is a value that's pretty close to 1. However, when they're abstinent, they are significantly underestimating time. So it's as if time has speeded up when they're abstinent. Okay, so this is an index of, they thought time was passing faster than it actually was. And then in the second study, we saw a very similar finding. So we see a significant abstinence effect. Not sure why, but these folks were not quite as accurate as the first group. But we see a similar effect where the direction is in the same direction, where time has speeded up when individuals are abstinent. We also gave them a standard delay discounting task, so asking them to make choices between smaller immediate rewards versus longer term but larger rewards. This is an index of impulsivity. And we were interested in whether there would be differences between an abstinent state and a smoking state. And we hypothesized that people would make more immediate choices. And we saw no abstinence effect across the 2 sessions in either study. So we see very similar effects in both of these. So in these samples, abstinence was not affecting decision making. And I don't actually, is this something that you've found in terms of like state changes in discounting?
Yeah, so it's a variable, and most of when there's very low magnitudes, perhaps like you have here.
Yes. Yeah.
There seems to capture less.
I see. Yeah.
Effects of different interventions.
Yeah.
So we tend to always use $100 or $1,000.
Okay.
To capture our effects.
I see. Okay. Well, for the next study then.
Absolutely.
Yeah. And so what we were interested in is the change between abstinence and smoking and how that's related to the ability to quit smoking. And so our outcome here is the number of days that people were abstinent during our practice quit attempt, and we also looked at the total number of cigarettes that they smoked during this quit week. And what we saw, so these are all of the different measures that we looked at, the only significant predictor of relapse was delayed discounting. And so this is the change score between abstinence and smoking. And so the next graph I'm going to show explains the direction a little bit, because the direction of this effect was a little bit surprising to us. Because you would expect that individuals who got more impulsive when they were abstinent for fewer days, right? They would have more difficulty quitting smoking. What we found was that individuals who were more impulsive during abstinent compared to smoking, so they were more likely to choose immediate rewards, were actually smoked fewer cigarettes during the quit week. They were abstinent for more days. Now, this was puzzling to us because it goes contrary to pretty much every finding in the literature. But when we thought more about our paradigm, what we were doing was we were incentivizing abstinence. So we were giving people small monetary rewards for each day that they were providing biochemical verification of abstinence. And so for somebody who is more likely to choose a small immediate reward when they're abstinent, they're valuing that small immediate reward right now, and so they might be willing to refrain from cigarettes to get that small immediate reward. And so what we would like to do in sort of a future direction is look at different schedules of reinforcement in a contingency management model, and whether we can tailor those schedules according to measures like this. So using delayed discounting is kind of a tailoring tool to identify individuals who maybe show this pattern. Maybe they get smaller but more frequent rewards during a contingency management treatment, whereas individuals who show no abstinence effect or maybe have lower discount rates might do just as well with more longer-term spaced out rewards. And so that's kind of a future direction, because we weren't able to test that in this study. The other thing that we were interested in was, how is time perception related to delay discounting? That's how we were interested in the first place. Is time perception kind of a mediating process of delay discounting? And in this study, we didn't see any correlation. And I think one of the reasons for that is our time perception task that we used, the temporal measure was on the order of seconds. Right? And our delayed discounting measure was on the order of days, weeks, months. And so I think that it's difficult to identify a correlation between measures that are so temporally different. And so again, as I mentioned, this is something that we would like to evaluate as a clinically relevant measure for tailoring treatment in the future. Okay, and so how do we translate this intervention? And so we've showed that cognitive deficits during abstinence are important, and particular groups of people might be predisposed to cognitive deficits, and so different strategies to enhance cognition. So the next couple of studies I'm going to talk about are two different strategies that we've tested to enhance cognition. One is a pharmacological approach. The other one is a non-invasive brain stimulation approach. I talked a little bit about the contingency management. And then the other aspect is, so we did show that the time perception is altered during abstinence, and so it might be useful and important to use techniques that can expand that temporal horizon when somebody's experiencing withdrawal symptoms. Okay, so before I go on to the next set of studies, any questions or anything about any of them? Yeah.
Yeah, I'm not sure necessarily how related this is to your study, but we're talking about like delaying reward. Something in mind was like the marshmallow test with children.
Mm-hmm.
I didn't know, or the question that came to mind after that was whether this kind of behavior or this sort
Yeah, that's a great question. So I think, so at least my understanding of the kind of trajectory of delayed discounting is that younger individuals show steeper discounting curves, and I think that it probably peaks in adolescence, and then as we age, we kind of age out of delayed discounting. So for example, we often see correlations between age and delayed discounting, and in terms of how young you would see that, I would have to look that up. I'm not too familiar with sort of the developmental process of that, but I'm sure there's some folks in the room that could probably speak to that. Okay, so the next study that I'm going to be talking about is an area of research. So I have 3 different studies where we're looking at, it's a repurposing medications model. And so what we're interested in doing is taking medications that we know they're FDA approved, so we know they're safe, we know their general target mechanism, and whether we can repurpose them to target specific symptoms of nicotine withdrawal. And the reason for that is developing new medications takes upwards of 10 to 17 years and can take billions of dollars. Most of them fail in very early testing phases, and so this is a more efficient way of developing more available treatments for nicotine dependence and other indications as well. Okay, and so why were we interested in, so the first set of medications that we were interested in were acetylcholinesterase inhibitors. And this came out of some early research from a former postdoc in our lab who showed that polymorphism in the choline acetyltransferase gene was associated with smoking relapse. And it turns out that there's no medications that target choline acetyltransferase. However, there are medications that can enhance endogenous levels of acetylcholine, because that's essentially what we were interested in doing, and those are the cholinesterase inhibitors. And they are approved treatments for treating the cognitive symptoms of Alzheimer's disease. So these are medications that can slow the progression of those cognitive symptoms. And they work by inhibiting the enzyme that breaks down acetylcholine in the brain. So it essentially enhances endogenous levels of acetylcholine. They improve working memory in both patients and volunteers. Some of our preclinical collaborators have also shown that they reverse cognitive deficits in rodents. So in these contextual fear conditioning, which is a measure of hippocampal dependent learning. And so we before we moved into our human study, we asked some of our preclinical collaborators to see, let's test these out in some of our rodent models first. So we have some collaborators who are doing preclinical models of nicotine self-administration in rats. And so Heath Schmidt was really the pioneer of doing this work. And so this is a great example of kind of the bedside to bench approach. So we have this clinical model, we have this hypothesis, can we test this in our animal models? And so what Heath did was he evaluated whether galantamine, which is a cholinesterase inhibitor, whether it would attenuate nicotine seeking in rats. So the rats are trained to self-administer nicotine, they like it, it's reinforcing, and when we pre-treat them with galantamine, what we see is a significant reduction in the number of times that they press to get an infusion of nicotine. And so when they get the placebo, they're pressing upwards of 100 times in about a 2-hour period to get infusions of nicotine. And we can reduce that to about 50 times in that 2-hour period with galantamine. The other important piece of this is that one of the downsides to the cholinesterase inhibitors is that they can have negative effects on GI symptoms, so they can often cause things like nausea and vomiting. One of the things that I've learned working with Heath is that rodents don't vomit, so it's just not a thing that they do, and you can't ask them how they feel. And so the way to assess whether a rodent is not feeling well is this phenomenon called pica. So when they're not feeling well, they will eat non-nutritive substances, and it sort of helps to coat their stomach. And so the way that we do that is give them kale and clay. It helps coat their stomach, and so the amount of kale and clay that they eat is associated with how terrible they feel. And so if we saw that they were eating significantly more clay when we gave them galantamine, this would suggest that maybe they weren't self-administering nicotine because they didn't feel well. And so what we see, there's no effect. Right? So they're not changing how much clay that they're eating. They're not changing how much food that they're eating, so we're not affecting other reward systems. So it's suggested that it is specific to nicotine seeking.
How about other goal-seeking behaviors motorically? Just when they're on the cholinesterase inhibitors and you have them pressing a lever to, get something else, for example.
Sure. In terms of like
Any effect?
other substances of abuse, or just or
Just
locomotor
more
activity?
general locomotor, yeah, that's
Yeah.
right.
So in the chamber, I don't have the graph up here, but there is a non-active lever, and to control for that. And so we don't see changes in the number of times that they press for the inactive lever, suggesting that it's not changing that sort of more general locomotor activity. So these effects were promising to us. It was suggested, well, okay, now let's carry out our human clinical study. And what we see, so these are data from my K award. The project is still ongoing, so these are sort of preliminary data, but the data are promising. And so what we see is after these smokers have been taking galantamine for 3 weeks, we see a significant reduction in the number of cigarettes that they're smoking, and this is before they've even quit. So we haven't asked them to quit. This is during the run-up phase. So this is a time in which we've said, just smoke according to your urges. Just follow your urges. We're not asking you to change your behavior yet. And they're still smoking significantly fewer cigarettes than the placebo group. We also see significant reductions in self-reported smoking satisfaction and how rewarding they're finding their cigarettes. So how much do they like smoking? And in addition, while we see some bumps in side effects in the galantamine group compared to the placebo group, if you look at this scale, so this scale for our side effects goes from 0 to 3. So we're seeing about a 0.1 increase in side effects. And importantly, these side effects weren't related to smoking behavior or other subjective responses. And so we don't think that the effects that we're seeing are because our smokers aren't feeling well. Okay, so we've shown some promising effects with our cognitive enhancing medication. One of the other approaches that we're very interested in using as a potential therapeutic to enhance cognitive function is non-invasive brain stimulation. And so the techniques that we've used are transcranial direct current stimulation. So these are light currents applied to the scalp, about 1 to 2 milliamps. And we are particularly interested in targeting brain regions that we know are important in executive function, like the left dorsolateral prefrontal cortex. And they've shown some initial efficacy in the treatment of other neuropsychiatric and neurodegenerative diseases. And it's very safe and well tolerated. And so this is a study, so this was from Karen Lerman's lab, showing that, I'll just skip right ahead just in the sake of time here, but so what we showed was that we can, using active stimulation, we can increase the latency to the time that somebody smokes their first cigarette, so which is an important indicator of the ability to quit smoking. So if you can increase that time, the more that you can increase that, the more likely is somebody will refrain from smoking. And it also reduced the number of cigarettes that somebody smoked during an ad lib period. Right? So it looks like transcranial, the TDCS, can affect smoking behavior. And so in the next study, I wanted to know, can we use this technique to modify other health behaviors? And so for this study, we were interested in eating behavior. And in particular, we were interested in, can we train inhibitory control to reduce eating behavior, to reduce chocolate consumption specifically. So this was a pilot study. We wanted to know if we can detect some signal in a very sort of narrowly focused outcome. Then we would sort of expand it to a more broader outcome. By stimulating the right inferior frontal gyrus, which has been known to be important in inhibitory control processes. And so we had our participants do what we called a chocolate go, no-go task. And this is a task developed in the lab to train inhibitory control. So half the participants in this study were trained to not press, so their no-go cue was always associated with an image of chocolate. So this way they're saying, every time you see an image of chocolate, you're not pressing. So we're trying to train the response to not go during a chocolate cue. The other half of the participants, the no-go and the go cues were 50% for the chocolate. So half the time they were pressing for chocolate, half the time they weren't. And what we wanted to know was, can we enhance the effect of that training using TDCS? And then everybody got an ad lib chocolate consumption task where we put them in a room with 3 bowls of chocolate and we say, we want you to just taste each of these bowls, eat as much or as little as you want, and we want you to rate them. And folks had been, so our primary outcome is how much chocolate they eat during that period. And so we have folks in the lab for about 3 hours before they do the task, because we wanted to standardize kind of their hunger levels and how hungry they were when they were doing the task. And during they're kind of hanging out in a lab reading magazines. We also had them do a set of baseline inhibitory control tasks, including a delayed discounting task. And we focused specifically on females for this study. Females in general are more likely to crave and eat chocolate. We selected individuals who reported that they craved chocolate, so we had a series of questions to ensure that individuals would be chocolate cravers. And we also standardized when the sessions occurred in menstrual cycles. There's some evidence to suggest that craving levels can fluctuate during menstrual cycles, and so we had everybody come in during the late luteal phase to control for that. So these are the results. So we didn't really expect training performance effects on our training task, but we see that in general, folks did pretty well. This is exactly what we would expect to find. We didn't see any differences in the active versus the sham condition. Now for our ad lib chocolate consumption, so remember what we're trying to do is inhibit chocolate consumption. What we saw during the active, we see a significant increase in chocolate consumption. Now, these data are very fresh. I analyzed them over the weekend, so I'm still sort of sitting with them and trying to determine, exactly what the meaning is. But what was really interesting about this is, so this was puzzling, and so what we did was we looked at, well, in the chocolate no-go condition, this effect goes away. We see no effect of the active versus the sham condition. So these are the folks who were never pressing for chocolate. The effect in the folks who were sometimes pressing for chocolate and sometimes not, this is where we see the largest effect. And it's a difference of about 13 grams, which doesn't seem like a lot, but when you think of it, it amounts to about 70 calories worth of food in about a 10-minute period. So it's a fairly large effect. And so right now what we're doing is we're sitting with this and trying to think about, where do we go next? What is our next step with this? And in thinking about it, so delayed discount, so before I get into that, so baseline delayed discounting was associated with chocolate consumption, which is what we would expect. So individuals who were steeper discounters ate more chocolate during the ad lib session. And I'm going to just skip through this as well. So we saw effects in our smoking study that were consistent with our hypothesis, but not in our food study. And so then we started to think about some of the differences between the two studies. So we were stimulating different brain regions. So it's possible that if we had stimulated the left DLPFC, we might have seen different effects. We also had individuals doing different tasks. So during the smoking study, while they were getting stimulated, all they were doing was trying not to smoke. So they were not doing a cognitive task. They were sitting there looking at their cigarettes in the room, and all they were focused on was not smoking. Where we had individuals here doing this inhibitory control task, and so there could be some effect of, if we had done a very similar paradigm in this study, we would have seen similar results. So just in the last few minutes, so to get to the sort of last component of the work that I've been doing over the last few years, so this is focused on HIV as a comorbidity in smoking, and specifically targeting cognitive function as a risk factor for smoking behavior and smoking relapse in this group. So over the last few decades, with the advent of antiretroviral therapy, the primary cause of death in individuals with HIV has shifted to non-AIDS related diseases. So they're more likely to die from cardiovascular disease, from cancer. They're more likely to get metabolic diseases. And in fact, recent epidemiological evidence suggests that they lose more life years to tobacco use than they do to HIV. And so this is a significant problem that needs to be addressed in this population. And so as I mentioned, they're more likely to smoke. Also, what's important is that our current treatments, like nicotine replacement therapy, are less effective in this population. And so we need different and new available treatments for them. There's a lot of different comorbidities that could potentially explain the high smoking rates in this population. So depression is highly comorbid in this population, and we know that depression is also associated with smoking and difficulty with the ability to quit smoking. And then there's also HIV-associated neurocognitive disorders. So these are deficits that are a consequence of HIV infection. And since the advent of ART, the prevalence and the progression of HAND has decreased substantially, but it's still associated with significant functional impairment. And as I've talked about before, cognition is an important risk factor for smoking relapse. And so we were interested in the intersection of HIV and cognition and smoking. Okay, and one of the reasons that we were interested is the domains of cognitive function that are important during smoking withdrawal, sustained attention, working memory, processing speed, are also very similar domains to what we see in HIV-associated neurocognitive disorder, or HAND. And so there's a lot of similarities here. And so what we wanted to know is, do smokers with HIV exhibit worse task performance on these cognitive measures than smokers who are uninfected? And so we had a clinical trial. So this was Robbie Schnoll's clinical trial, where he was recruiting smokers with HIV for a placebo-controlled trial of varenicline. So these are treatment-seeking smokers. And then I had the sample of smokers from my K award, who are HIV uninfected smokers, also treatment seekers, also enrolled in a medication trial. And what's important is that all the smokers in both of these studies completed the same cognitive tasks. And the inclusion criteria were very similar across both of the studies, and we evaluated cognition prior to the initiation of any treatment. And so we were able to evaluate baseline cognition and compare the two groups. So these are the two tasks that they did. So the first was a working memory NBAC task. So this is very similar to the one I showed you before, except it's a visual spatial task where they're asked to remember the location of this dot as it appears around the screen. So it's a little bit of a different take on working memory. And then they did a continuous performance task, which is assessing vigilance and attention. And these are so we characterize cognition using these three different metrics. So for each task, we assessed accuracy, processing speed, and we're also interested in inter-individual variability as a measure of consistency in your responding, and can also be considered as sort of lapses in attention. And what we found was that between the two groups, working memory accuracy, working memory reaction time, and intra-individual variability on our continuous performance task were all significantly different between the two groups. And in all cases, the HIV-infected group performed worse. So they were less accurate, they performed more slowly, and they were more variable in their responses. And so we wanted to, well, is this unique, right? Are we capturing something else that's different between the two groups, whether it's socioeconomic status, education level, other types of substance abuse history. And so we conducted an ROC analysis to evaluate whether we could enhance our ability to classify the two groups with our cognitive predictors. And so this red line here is where we included only our demographic variables and our smoking-related variables as predictors. And so when we're trying to classify HIV status, we do pretty well with just those variables. We get about 70% accuracy, so we do okay. When we add our cognitive variables to the model, we do significantly better. So we get about 85% accuracy. So it suggests to us that we're capturing something unique about the populations that is different between the two groups. Okay, so we showed that the smokers with HIV, they perform worse on our measures of working memory and attention. And then when we looked specifically at our population of HIV-infected smokers, we also wanted to know, how does cognition relate to some HIV-related outcomes? So how does it relate to things like viral load or their CD4 count, things that are important predictors of their health and medication adherence? And what we found was that it's associated, so lower working memory was associated with higher depression scores. This is something that we would expect to find. It's also associated with lower self-reported quality of life. So individuals who perform worse on the task reported just general lower health-related quality of life. We also reported, we also found that lower working memory was associated with a higher CD4 count, which is somewhat counterintuitive. We would have expected lower working memory is associated with lower CD4 count. We want higher CD4 counts are better, indicate better health. But it turns out that current CD4 count is not always the best index of health. What we want to look at is the nadir CD4, which is essentially the lowest level of CD4 count that they have. And that's a much stronger predictor and is much more strongly related to cognitive function than current CD4 count, because that can fluctuate a lot more. So there's some possible neurobiological mechanisms. So HIV infection can have effects on neuronal damage and has been shown to affect some of the same brain regions that are important in cognitive function, like dorsolateral prefrontal cortex and the medial temporal lobe, which is important for things like hippocampal dependent learning. And then in some of our follow-up work, which I'll talk about later during the Chalk Talk, we're interested in the role of persistent inflammation as a contributor to some of these cognitive deficits and the interaction with inflammation and smoking. And right, so that's kind of the, if you have time at 4 o'clock today at the Chalk Talk, these are some of the studies that I'll be talking about. And I think I'll end it there. So these are acknowledgments. Again, this couldn't be done without all the team, and then all the funding agencies who've made the work possible. So if you have any questions, I'm happy to take them.
So in your later studies, you're talking about, is there a baseline difference in cognitive function and how does that interact? If you go back to your earlier studies, if you measured baseline cognitive function in just plain smokers, would there be any systematic relationship with the withdrawal-induced cognitive dysfunction?
So in other words, so if folks have better cognitive function at baseline, is there a difference in how much they're affected by withdrawal? So we've seen that in some cases. So the individuals who show stronger cognitive function at baseline are often most affected during withdrawal. And I don't know if that's because because of floor effects for if folks who have poor cognitive function at baseline, if they just can't get any worse during abstinence. But yeah, we tend to see that individuals who perform better at baseline show the greatest deficits. Yeah.
So towards the last part, you started to get to this about the possible mechanisms of
Yeah.
HIV. It occurs to me, you may have scratched the surface of something telling us about a side effect of the retrovirals. That is, if you're getting a very positive effect on CD4, it could mean that you're sensitive to more sensitive to the retroviral treatment, which could mean you're more sensitive to it having other effects. So in other words, instead of thinking whatever's happening is downstream from your CD4 counts and all those sort of things, it could be some other totally unrelated effect of the retroviral treatment that's causing this deficit in working memory.
It absolutely could be. And I think that there's some evidence to suggest that depending on the type of antiretroviral that people are taking, it can have effects on things like metabolic function, which could potentially have, other effects on inflammation and cognition. There is one antiretroviral, efavirenz, which has shown some association with cognitive deficits. And I don't think people really fully understand why that particular medication is associated. We didn't have enough people in this study to really tease that apart. We just didn't have enough people who were taking that medication. But yeah, I don't know that people have really done enough work in that area to identify why antiretrovirals might contribute to some of these effects.
And in corollary, just quickly, I should know this when you don't. If people are being treated for depression pharmacologically, not HIV related, other reasons for depression, does that improve their working memory performance generally, no matter what the source of the depression is?
No matter the source. So there is some evidence. So I think it also depends on the class of medications. So there's some evidence that bupropion, which was originally an antidepressant, was remarketed as Zyban for treating smoking cessation, that can have some cognitive enhancing effects. To my knowledge, though, I don't know that there's been any systematic studies looking at that. I think like some of the tricyclics actually show potentially negative effects on cognition, at least the little bit that I know about it. Sure. Yeah.
You mentioned that HIV-infected people are more likely to smoke and also more harder time getting the treatment effects. And I'm wondering, one of the cognitive mechanism that can explain is their time perspective. That because they expect that I'm not going to live that long, like just
yeah.
The horizon is so reduced that
yeah.
it may affect their attitude.
Yeah, it's funny. Warren and I actually talked about a little bit about that yesterday, and sort of wanting to do some of the episodic future thinking, particularly in this population, right? If they have a shortened time perspective, I think I think that would be a great study to do. Yeah, absolutely.
Great. Well, thank you very much.
Sure. Thank you for coming.